Welcome back to post-lunch session of National Webinar 2024 on Advancement of Nanotechnology in the Treatment of Reproductive Tract Diseases, a journey from bench to bedside. Organized by Smriti College of Pharmaceutical Education Indore in association with Indian Society for the Study of Reproduction and Fertility. Moving ahead, we are very fortunate to have among us Dr. Neelesh Kumar Mehra sir, Assistant Professor, National Institute of Pharmaceutical Education and Research, Hyderabad. So I request Mrs. Manisha Pere Madam to please display sir's profile slide. Dr. Neelesh Kumar Mehra sir is working as an Assistant Professor of Pharmaceutics at the Department of Pharmaceutics, National Institute of Pharmaceutical Education and Research, Hyderabad, India. He earned his PhD from Dr. H.S. Gode University, Sagar, and postdoc from Irma Lerma Rangel College of Pharmacy, Texas, USA. He served as manager in product development, Centis Research Center, Centis Pharma Private Limited, Gurgaon, for development, scale-up and technology transfer of complex ophthalmic inhalation and optic pharmaceutical products. He received Team Award for successful commercialization of ophthalmic suspension product. He has authored more than 120 peer-reviewed publications in high-repute international journals and more than 30 book chapter contributions. He has granted 4 patents on manufacturing process and composition to improve therapeutic efficacy for topical delivery. He is guiding 14 PhD and guided 35 MS students for their dissertations research projects. He has received numerous outstanding awards, including Young Scientist and Team Awards for his research output. He is a peer reviewer of various international journals and publishers. He recently published 3 edited books on multifunctional nanocarriers, biopharmaceutical, and nanotechnology. He has rich research and teaching experience in the formulation and development of complex, innovative ophthalmic and injectable biopharmaceutical products, including micro and nanotechnologies for regulated market and so on. According to announcement by the Stanford University scientists based on the database and basis of standardized citation indicators, Dr. Mehra's name is included in top 2% scientists in the world in the area of pharmacology and pharmacy consecutively for 3 years. So, we welcome Sir for his insightful talk. Sir, now you can share your screen.
Hello, is I am audible? Hello, okay. Thank you for confirmation. So, thank you for giving a brief introduction about me, and I would like to thankful to the entire committee for inviting me to give a talk on this advancement and the recent trends in the drug vaginal or reproductive drug delivery system or the drug delivery, the different diseases by which we can be used to treat the different reproductive product. Just wait a minute. Yeah. So I hope this slides is visible for everyone.
Yes, sir.
Yeah. Thank you. So today's my topic is recent trends in the mucoadhesive drug delivery system. So I would like to add on that the most recent trends in the drug delivery system, which could be helpful for the treatment of the different reproductive drug disorders or the diseases that is associated with the reproductive system. So first I would like to disclose the disclaimer. The presentation which I had covered it, the views that is reflecting of the views of the presenter, that's me, and it should not be any constructed to the represent of any views or the policies of the Hyderabad. The information which I have taken from the various sources, it includes the scientific journals it has the published data, and some data we have added that is unpublished data from my lab. Those are working in this particular area. And I had taken some data from the regulatory guidance, the different literature which is available in this particular area from the Google. The specific guidance and the product data is taken from the different regulatory agencies. So I just give you the one quick look on the evolution of our entire drug delivery system. This is you can see the slides. It has been cover up of the entire history, which is if we talk about the long back history of like 1950. So we have been started to look on the particular on the dissolution control and followed by the diffusion control. So that's the one advancement or the trends was done and particularly in the dissolution and diffusion based mechanism. So where over there, the Spansul and the Occusers kind of the dosage form has been successfully launched, and those are all available in the market nowadays. Followed by like this osmosis, the mechanism has been taken, and the Oros, the one product which is available in the market again. So this is like the initially we had work on the dissolution and diffusion mechanism based product development, and then followed by the osmosis process. So later on, like if we talk about like 1990 segments, where the new things or the novel drug delivery system has been came in the picture. So firstly, we had done the research, the investigation or the innovation work has been done on the microtechnology. So the microtechnology, the first product like this one, you can see it in 1990s, particularly in 1989. So the one product which is based on the PLGA microparticle. So this is the one which we can say that the microtechnology has been came in the picture. So the Lupron report, this is the one innovator product which is available in the market, and now in our India also so many companies are trying to develop of this Lupron reports generic product. So this is the one where we had started to work on the microtechnologies, and later on we switch on from micro to nanotechnology by coming in this nanomedicine era. So the nanomedicine is nothing only from switching from micro to nano, and wherever we had to work on the particle size or the vesicle size, where from that would we come in the nanometric size range. So we started from like 2002, 2010, wherever the work started on the particle, this nanoparticulate technologies, and we know about that the success story, the product which is available based on this one, like the Abraxane or this Taxol, which is like a paclitaxel in the pegylated castor oil. Abraxane having the like paclitaxel bound with the albumin complex, particularly human serum albumin. So those are having the like particle size 130 or nanometric size. So this is the glimpse I was just want to be show here, that we had like starting in the different era with the different technology. So there is always a scope for that to do some kind of the innovation, some kind of the new research onto the existing marketed product also. So there is also, we can say like that this is like some Abraxane paclitaxel complex product is available. So how we can change it, the products, what the problem is associated with the Abraxane itself so far, so how we can mitigate. Similarly, like Doxil, which is also the pegylated one of the liposomal formulation. So they have also some problem. So or some challenges. So those is we can be sorted out or we can be mitigate also. And then nowadays we have been like 2020 to 20 to 2030, wherever we has been come in the picture of like that we should be work on to the complex product development, by which we can be deliver of some peptides or some biologicals, drug substance or the biological compounds, and also we have been looking care of some product that could be work for the long-term treatment and the product is like in a nanotechnology field. So this is I just had cover up from 1950 to 2030, and so many products are available in a market which is based on the micro as well as nano particle weight also, and the some few are under in the pipeline in the clinical trial phase 3 and phase 4 also. So now coming to the topic, which is the based on to this today's webinar, so that I had covered up some diseases or the disorder which is related to the reproductive system, particularly in the female reproductive system. My lab has worked on some 2 to 3 projects, so they so that it would be beneficial for the human healthcare society or human benefits. So we talk about like this the vaginal drug diseases. They are basically like infectious diseases or the cancerous diseases. You can see is like infectious disease. The infection has been come from like trichomonas, bacterial, different kind of the bacteria. They may be impacting onto the those particular vaginal area. So like a bacterial vaginosis is there, like candidiasis, wherever the candida species may be impacting onto the, and they create like some kind of the infectious diseases. Now coming to the cancerous diseases like that, where the cancerous has been developed. Initially, it has been infectious, or it in the primary stages. Later on, it could be developed of the cancer in a different stages also, like this one. So the we talk about like the composition. Because like in case of the mucoadhesive proper drug delivery system, that is very recently trends. So many companies, even though so many research institutes, they are working on to the mucoadhesive drug. So why mucoadhesive is like that in the particularly reproductive? We know about the mucus has been present in the different body parts. Like it would be these kind of the drug delivery system would be helpful in the ophthalmic drug delivery ophthalmic segments. So where we had done also the mucoadhesive drug delivery system, they could be helpful in the ophthalmic segments. They could be helpful into the oral drug delivery system where they have interact with the mucin. So you can see it, the mucus, which is if we talk about the composition, the mucus has been almost 90 to 80% is comprising of water, glycoproteins, electrolytes, cell components, fats, proteins, enzymes, and immune factors. Almost one two. So the majorly is companies water, and the defense. If we talk about the defensive mechanism, the different kind of the barrier system is available onto they restrict like in terms of dynamic or static barrier or interactive barrier. So they are always interacting, and they are the one main challenging property. So they could be we have to be solve, or we have to mitigate these, that the drug has to be by crossing up this kind of all various system which is impacting on that. So for that, due to this composition, defensive mechanism of mucus, and like that, the engineering mucus barrier goes like that. So like some mucoadhesive particles, and how after that we have been transformed into the mucose penetrating one. So that just for the changing of some composition and some API excipients, inactive compounds we have to add. So their property has been changing out, and they are more capable to go to the penetrating into the mucus. So the one more important thing is the zeta potential altering. If you are changing the property, if you are altering the surface charge on the nanoparticles, so that they could be interact with the mucin, and they would be become like a muco penetrating kind of the particles also. So those kind of the strategy we could be apply here, not only in the reproductive system, or we can be apply in the ophthalmic segment. In my lab, sorry, we are basically dealing onto the ophthalmic segments and the topical and the oncology. So these are various physiological variables which I had talked on the previous slide also, but here you can see it, the most important in case of the vaginal drug delivery system is the pH. Nowadays you have those some product which is available. So they have only, they are nothing only, they have only 2 to 3 excipients. Those excipients are very common, and only what they will be do it, they are changing and they are controlling the pH of the vaginal pH. So like for example of VWAS, like this one. So this is only that what the strategy or approach the product company has applied to change only that. If in a certain kind of the infectious infection disease or the some infection has been occurred, then the pH is the one primary important factor that has been altered. So only that to regulate the pH, it is only the one. So that's for that the important thing, aspect is that the osmolarity, not only in the mucoadhesive one or vaginal field, the osmolarity is always play in a role in a vaginal drug delivery or mucoadhesive and the ophthalmic segment also. The different kind of the various system we have to be take care, and we have to be design the product by the composition, by changing in the pH, by changing in the osmolarity, viscoelastic, and some different dynamic or properties of the product, so that they could be more compatible in that pH condition, so that the product has to be deliver the drug compounds in that particular area for that disease. Enzymatic degradation is also there, so that we have to be take care. Epithelial changes due to the this menstrual cycle period, so that those are conditions which is very critical in case of this vaginal drug delivery system. So that's why we have to be take care for the adhesive one, mucoadhesive one, or the mucopenetrating. Now coming to the vaginal mucoadhesive drug delivery system, the some kind of the mucoadhesive drug delivery system, you can see it here in the right side. So there is not only like I can say the ophthalmic one, we can be do it the innovation or the research by with the help of mucoadhesive one, ophthalmic in situ kind of the forming gels or the eye drops we can be prepared, or some kind of the micro needles or the micro or needle waste kind of the films or the lens we can be prepared, the different kind of the biodegradable lens we can be prepared. So this is in case of the ophthalmic. I can say also that it is related to the oral drug delivery system, that this mucoadhesive we can be apply for the nasal also, pulmonary treatment also, and the vaginal one also. So this is like you can say the buffer gel which is available in the market. That is the gel which is comprising of the carbopol. That is the polymer. We know about the different kind of the viscosity grade is available, and accordingly we have to be prepared and by selecting of the particular type of the viscosity. So that could be work as a mucoadhesive agent. This carbomer is having the good unique properties. They could be helpful to develop of a gel or to develop of this in situ gel also in the ophthalmic and other segment also. So that's a unique polymer which we can be utilized into the this one in a mucoadhesive drug delivery. So on the left side, the different kind of this for drug delivery system, they are available like in the tablet or the capsules. Those products are available in the market. Creams are also available in the markets. O-rings are available, suppository, syrups, injectable. So these are the products which you can see this side. They are available in the market. But I had previously in the previous slide that there is always a scope to do the some changing or some improvement in the existing product, so that they could be improved or they could be show the better improvement in the therapeutic efficacy. Or nowadays because it's a world of nano, and we are dealing with a different kind of the nano carrier system. So like a polymeric based system, polymeric mices, dendrimer also, the liposomal polymeric complex also, or the polymer drug conjugates also. With these the different kind of the nano carriers, they could be helpful for into the treatment of this vaginal drug delivery disorder also, and they could be more helpful to treat the cancerous disease which is related to the reproductive system also. So we know about the statistics if we talk about the particular any case of the cancer. So that you can see it here, the different types of the cancer, their incidence and the mortality rate and the rank. So the cervix uterus, this is the one having the 8th rank, and the corpus uteri which is having the 15th, and the ovarian cancer, and followed by the vulval as well as vaginal cancer also. If we talk about the mortality, you can see based on that, we have been seen it that the one vulval, which is having the 50% of the mortality one. And if you see the demographic of vaginal cancer in the particular incidence, so Asia having like more highest prevalent into the vaginal cancer, like the Asia 54% is showing. After that, Africa 11%, then Latin America and that 8%, and North America, and the second one is the Europe. So this is the incidence which is have been related to the vaginal cancer as well as the mortality, the 57% are in Asia also. You can see the different color coding by this one, the statistics also. Sorry. Yeah. So now coming to the another factor that we want to be understand about that, the risk factor for this carcinoma or the cancers. The risk factor we like age, previous any like this neoplasmic or of the any condition which is related to the cervix and the like this any other diseases which is like a STD one. Other risk factor like gynecological dysplasia and neoplasia. The smoking is also one factor that is associated. Low socio-economic status like in African countries also, previous pelvic radiation also, unstable marital status, number of sexual partners, younger age at first intercourse, vaginal trauma from fisheries, flops, multigravid and hygienic factor also. And these are the recently we had cover up the possible risk factor that is also sometime has been reported all immunosuppression, estrogens, older age at the younger age at in case of the menopause or any other malignancy, that is always is cover up into the possible risk factor to create or the develop of this cancer in a female reproductive. So that's the just brief about of the initial introduction about that how the drug delivery system would be helpful and how the different kind of the cancer as well as the infectious disease has been come in the female reproductive disorders or diseases. So I had covered up some few case studies which would be helpful to understand about that how the research has been going on to treat of and these kind of the different kind of the disorders or diseases. So that you can see the scientists they have developed a temperature sensitive gel which is comprising of the cyanovolfragan and resivolfragan in this particularly with the help of solid lipid nanoparticle. So we know about the solid nanoparticle, they have the capability, they can be encapsulate the both lipophilic as well as hydrophilic drugs depending on to the how much the drug they could be able to load it. The loading properties depending on not only in the solid lipid nanoparticle, that is depending upon to the properties of the API also. So they had loaded these two and they obtained from the toad venom and real gun. So they are showing that initially what we will do, like in this case, what I say that these are the compounds which have been obtained from toad venom and the real gun. So initially what we have to do, we can check it out on the cervix cancer or the any this if we know targeting in the different cancer. So they target particularly cervix. So what we will do first step, either we will be check out through cell line studies that either this compound would be helpful to treat in this one and we calculate the IC50 also. And after that, they are really helpful to do this activity or showing this activity. After that, they how much they will be do improvement in case if we are giving in a combination. So the two things we will be say. One thing, if we are giving the combination index we have to be calculate, and we will be see that either they would be synergism or also or not. And the second one, if the synergism is there, we call this combination in a fixed dose combination. So the FDC, fixed dose combination, wherever the product is comprising the two compounds or the more than two compounds, we call like a fixed dose combination. We should be see and tested and plan for the dosing in a combination with the fixed dose manner. So they had developed the thermosensitive hydrogel. You can see the picture. They are transmitting initially in the solution form, and when we subject it the change in the temperature from 25 to greater than 35 degrees Celsius, it has been form of the gel. So this is the property when we call like in situ gel or thermos, those are responsible with one factor, that is the temperature one. So initially they had developed the solution. Later on, while using, it will be form of a gel, and we have to be take care the property of the solution also, as well as we have to be take care the properties of the gel also. So how does it behave when we develop at a room temperature 25 degrees in a solution form, and once it will be given into the desirable temperature condition, they transform in a gel, and how does all the properties has been, there is some, the property is changing or not, or those things we have to be take care. Because why am I telling in an industry, because I had worked in the industry, I have seen it critically, it is not easy to say that the some properties they are forming, but we have to be generate the data, we have to be show to the regulatory agencies. So this side, you can see they have tested onto the radio labeling one, and the imaging has been done of this uh, both toad venom as well as the this solid lipid nanoparticles with a temperature sensitive in a nude mice. That's one critical studies by which we can be check it out that how much the product deliver the drug in a particular area of the segment where we want to be deliver the drug. This is also they had done the histopathological studies, and it is showing that. So the conclusion is that, and they had also checked it out in the cell cycle distribution in the particular HeLa cells as well as the ovarian cell SCOP3, which is known for the ovarian cancerous cell. And based on this study, they have been said that these two compounds could be helpful in a combination, and this combination could be used for the gel purpose, and so that the people who were suffering with this ovarian cancers, they could be helpful, and maybe the product will be reached into the market also. And they have also seen the toxicity. There is no toxicity has been found. The another which I had also done in the, which we had done with the mucoadhesive kind of the liposomal formulation, by which we have been taken up the chitosan that modify the liposomal formulation. You can see it. It is also published, and we had selected the carboplatin, which is one anti-cancer drug to treat the vaginal for purpose. So sorry. So in this, what we have been taking the phospholipid 19G cholesterol and the carboplatin, and we had been initially developed for this carboplatin loaded chitosan nanoparticles, and after we have been checked out the all physicochemical property as per the guidance, and we had seen it the stability also, so that the chitosan coated modified liposomal, which is comprising a positive zeta potential, that would be helpful into the treatment of the vaginal, which is comprising of the mucosa, which is having the negative charge. So the positive and negative interaction would be take place, and there is a chance for the more delivery of the carboplatin, by which we have been, you can see it here, the some parameter like particle size and the PDI and the zeta potential, and this TEM micrograph also, some electron microscopy. Another one, these are basically we do for all formulation. So this percent drug permission, by which we have been taking care, they got this uh, vaginal layer, and we have been seen with the help of French diffusion cells the percent drug permitted. You can see it, the CPLF1 and CCLF. So the planned formulation by which the chitosan is not there, and the CCLF1I, that is the chitosan coated modified liposomal formulation, which is showing the sustainable graph. So the drug or the gel which we can be applied like a once in a day, or so that could be able to release the drug for a 24 hours at least. We had subjected also the stability, because stability is requirement, and we know about that to check out the room storage condition also. So we have to be do the stability study for to maintain or to predict the shelf life. So these are the in case of mouse vagina which we have been done. One case study I had cover of that is related to a tenofil vaginal film. This is not from my lab. This is from the other lab. So that they have been done this for delivery of they prepared initially for the film vaginal film, and while loading of the tenofovir, that is you can see it in the A methyacrylic acid methyacrylic copolymer plus triethyl citrate and the tenofovir as a drug delivery system. So this is the one they had prepared the films, and that film would be helpful and to for improvement into the this tenofovir drug release pattern. So the main mechanism is involved in the diffusion, because this diffusion is the one process which by which this, and the second one is the muco adhesion one. So they had also checked out the different pH level, and initially what the role of plasticizer in this one. So this is the one technology that would be helpful, like in the form of a films also. So we could be helpful to develop this film. Not only like that's why I had initially told in the mucoadhesive kind of the film, which would be helpful into the ophthalmic segment also. The next one is the endometriosis. So this is another project in my lab. So endometriosis is a major contributor in the pelvic pain and subfertility, which is characterized by the endometrial-like tissue outside the uterus, like primarily on the pelvic peritoneum, ovaries, and the rectovaginal septum, and so this disease I have been selected to see because this is a cancerous one, by which we can, like in an endometriosis region, the growth of endometrial tissues on the sorry. Yeah. So that's the one status as the diagnosis stage for this endometriosis, and the prevalence of endometriosis in the female worldwide, like 41%, 80%, 51. The women and teenage girls with the pelvic pain have been suffering with this endometriosis, and the prevalence of endometriosis is higher in the women with the dysmenorrhea up to 60%, dyspareunia up to 44%, pelvic pain up to 80%. So we have been done this. I am escaping. So we had prepared the PLJ microsphere, which is loading of the one drug, Lagolux, and we have been optimized initially the polyvinyl alcohol is the different concentration from starting from 0.25 to 0.5 to 1%. And these are the beautifully same images of the PLJ microsphere. You can see it, the beautiful structure of the PLJ microsphere, which is the optimized one of the drug loaded one. This is again also 1%, which is showing the very smooth and porous surface structure of the microsphere. There is something happen. And we had also subjected for the in vitro drug release. You can see the zoom one is that the percentage cumulative drug release which we have been checking. The drug is the Elagolix, which only the one product is available in the market, that is in the tablet form. So always, as per like I just would like to add on that, if we changing from tablet to like a gel or any other form, like that would become in the filing stage of 505B2. So from where we can be changed from one dosage form to another dosage form. The another one which I have been work on that is particularly for the mucoadhesive chitosan nanoparticle. But here we have been utilized this modified chitosan nanoparticle, and we converted from mucoadhesive to mucopenetrating, as well as we have been seen it the comparison. How does the mucoadhesive as well as the mucopenetrating, which one is the best to deliver the drug into the particular ocular segment. But these kind of the modified chitosan nanoparticle we could be able to use for the this vaginal drug delivery system also. That's why I had cover up. So this is also we had done. We had published this one, and now we are checking out in the vaginal drug delivery system also, by which we have been loaded the different antifungal compounds also we are exploring, and these students they had worked on this particular projects, and we have been got the grants also. So the prevalence, this is related to the fungal keratitis. Yeah, yeah. So this slide is important, by which we have been check out the comparative between the mucoadhesive as well as the mucopenetrating. So this is only like that we know about the what does it mean by the mucoadhesive as well as the mucopenetrating. Only how does we would be change from mucoadhesive to mucopenetrating. You can see the schematic representation how does these nanoparticle like a mucoadhesive nanoparticle they would be helpful to uh, deliver the drugs by this crossing of epithelial cells and to the ground. And the once we have been changed the particles, the charge and we can be alter the surface charge. So the mucopenetrating particles, they having a particle size which is in a very appropriate particle size. They could be able to penetrate into the mucous pores, and so that the mucus penetrating surface helps to coating to prevent adhering to the uh, surface also. So by which we have been done this chitosan modified nanoparticles, by which we have been loaded with the some antifungal agents for this fungal keratitis. But those antifungal like fenticonazole and the voriconazoles and the samduliconazole we can be explored for the vaginal drug relief, which is for the fungal different kind of the fungal infection also. So this is the one is on the right side, the head cam assay, which is very important because this is the gap between to the in vitro as well as for the in vivo studies. So we have been if we do not want to be go for the in vivo study, we have been subjected for this permeability as well as for the treatment onto the with the help of this chorioelectric membrane that has well known and well reported into the literature also. So we had subjected on this both mucoadhesive as well as for the mucopenetrating one. You can see the cumulative percent drug release profile also, and this is the one mechanism how does from that the mucoadhesive as well as for the how does the drug has been came out and released out from to the gelling system or the drug delivery system also. The one more projects we have been working on to the treatment for the vaginitis. This is the one vaginitis. It's a common problem which occur really in with the help of this bacterial infection as well as fungal infection. If we have been like two different both pathogen like bacterial infection as well as for the candida species. So that mixed vaginitis we call it. So for this we can be explore like with the help of antifungal agents. So like once a product we have been developed, we are exploring against to the candida species as well as for the Staphylococcus aureus species also. So this one we had on that. So this is kind of the gel system we had developed, which is a nano. The size is around 120 nanometric size, and we had subjected to check out the interaction of these nanoparticle again between to the mucin one, so that the ionic interaction and how does the interaction would be helpful, or it is really interaction between the mucin as well as an nanoparticle has been take place or not. That we have been done with this one assay, and we check out. And we had done also the cumulative permeation study with the help of French diffusion cells to check out that how does the permeation has been taken care with the on the vaginal pH also. We had subjected the antimicrobial activity to see that particularly onto the two species. One is the Staphylococcus aureus, another one is the Candida albicans, and we had checked out that they are showing the efficacy at the lower concentration also. This is the one antibacterial activity as well as the antifungal activity shown on that. So those are the some studies which are working in my lab also. So that's why. And this is the one which we had a cover up and we published in the one article. So the available patent for vaginal drug delivery system by comprising of the different patents related to the different dosage form, including nanofiber, gels, nanoemulsion gel, in situ gel, nanoemulsion forms, and the patches and the capsule, including of the mucus penetrating nanoparticle also. So this is like giving a brief about that the product is going in the market, and they probably the US patent and this other intellectual property those having the these kind of the product will be come in the market in a later or in a future also. So the clinical evaluation for commercialization, whenever we are doing the some product, we have to think about the commercialization, technology transfer. So the key consideration for this, the development pathway has to be simplified and simplified manner, because the complexity is not easy to be handled in the manufacturing plant level. The second one is the safety and toxicity has to be checked initially into the in vitro, ex vivo, as well as the with the help of preclinical testing, and then we have to go for optimum concept for this clinical trial when we are going to launch of the any product in as a novel drug delivery system or NDA filing. So the challenge mainly we are facing, we know about the preclinical studies, we know about the safety and toxicity data has been required, we know about the clinical trial has been required, and we had learned, read it and through the literature or through the books. But the key challenges which the people facing, because we are aware the academic and industry people also, we don't know about the regulatory guidelines. We don't know, so that I give the one message, and my suggest to whoever the audience, their faculty or students, they must be go and read it all the regulatory guidance which is available on the particular type of the dosage form, topical, injectables, or the tablet. So they must be clarify all the regulatory guidance, how does the product has been developed. Like I just giving the one example, whenever we are developing one gel, do we know about this carbomer or the polymer, how much the concentration we have to add? The concentration that is available for the topical one, that is we should be take from the this IID database that is available on our website of the USFDA, particularly in active ingredient database. So why which we can be check it over how much the carbomer or the carbopol we can add in the gel in a case of topical. In case of the topical formulation for the gel, where we are taking for ophthalmic one, or if that we are developing of the tablet, that is the one. So that in the end, if you could just type it, you can just go on that one. You see the concentration which is already approved from the US FDA one. They are safe, and they are not toxic one. So that's why that's a kind of the lack of regulatory awareness or the guidelines people are facing. And the second one is the complexity of the patent and in the intellectual property also. So that must we have to protect whatever the product or the any innovation we are developing in the with the help in our lab. The future perspective, I would like to say that in the case of nowadays, the collaborative research work is more welcome. The people has to be work with the inter department, inter institutional, as well as the different labs. So the collaborative work research, because the one person is not like that expert in all discipline. One person or the one researcher having expert in the formulation, another having the analysis, another having the preclinical testing. So these kind of the collaborative research work will be giving the better product development or better dosage form. Complex formulation is always, because we have been forget about we should forget to the simple solution or any other products, so that we should shift toward to the complex formulation, and later on the translation, research, and then commercialization. So these are the aspects I would like to add on that for the future, so that we could be work it in this aspect. So I just cover up this slide. This is one slide, and the students are working under my supervision. Almost 40 students are working at the SRF and JRF, and almost 4 to 5 students they have been working on to the this DST, ICMR, and Inspire, and sub-granted project also, and the 5 students they are working and to complete of their dissertation work for the completion of the master MS degree. These are the 3 recently edited books by me. Then there are nanomedicine, micro and nanotechnology based product development, and the multifunctional nano career, and some are in under pipeline. These are the glimpses which we have been like. This is the one lab, and we have taken the photographs with my guide, Professor N. And this is the one photograph. Recently, I had organized the international conference on particular drug discovery, delivery, and these are the products which I had done, the prototype one, and they have been shown into the during the parliament within to the Sasi Tharoor, and this is the beautiful picture of my institute, my Hyderabad. Thank you. And last one, I would like to thankful to the organizing committee to give this chance to deliver the talk on this particular area. Thank you. If anybody have the question, they can maybe ask.
Thank you, sir, for your insightful talk on the recent trends in mucoadhesive drug delivery systems and reproductive diseases. Your expertise and passion for the topic were evident, and we greatly appreciated the depth of knowledge you shared. Thank you very much, sir.
Thank you. Thank you.
Now, I would like to request Dr. Rajiv Saxena, sir, to deliver valedictory remarks. Please, sir.
Thank you, madam. The time has come to draw a curtain to our program as we come close to it. On behalf of Smriti College of Pharmaceutical Education, Indore, and the Society for the Study of Reproduction and Fertility, I extend my heartfelt gratitude to all our esteemed speakers, participants, and organizing committee members for making the National Webinar 2024 on Advancement of Nanotechnology in the Treatment of Reproductive Tract Diseases, a journey from bench to bedside, a grand success. The inauguration session was viewed by 818 participants, and very soon the YouTube link part 1, consisting of inaugural session and first technical session, and part 2 having technical session 2, and part 3 having technical session 3 will be shared on your mobile number. We request you to please like and subscribe the video and YouTube channel as motivation. So now, if any participant want to share your view, the forum is now open.
Good afternoon. Good afternoon to all the participants. And I, Dr. Shaili Chaudhary, congratulate the principal, Dr. Neelesh Malviya sir. Sir, what a beautiful session you have arranged for us. And we are so thrilled to hear all the sessions. I must congratulate each and every member of Team ISCO for such beautifully, in a very beautifully manner, you have conducted this webinar. And we are fortunate to be a part of this webinar. Thank you so much, Neelesh Malviya sir, for providing us this platform to hear such a brilliant session of our speakers. Thank you, Neelesh sir. Thank you, Team ISCO. Thank you.
Thank you. Thank you.
Thank you, madam. I would also like to invite Dr. Sapna Malviya madam to share her views.
Yes, good afternoon, respected all faculties and eminent people over there. And throughout morning, we are in the session. It was an enormous pleasure, and we have learned lot of knowledge from this particular session on the advancement of nanotechnology. Indeed, we are thankful to all the speakers of this particular session, because we have learned a lot. Especially we have listened the session since morning. We have been involved. We have listened Dr. Lohia sir and then Shashank sir, and then we were very delighted to listen other speakers also. So it was an extremely useful session which has the brief overview about the nanotechnology. We were thrilled and we were excited to listen, and we have learned a lot. I again congratulate Team Spoke and Principal Dr. Neelesh Malviya ji for organizing such a wonderful session. And throughout the session, you have moderated very well, Dr. Rajiv Saxena sir, and all the team of organizing committee of Scope. It was very well organized. We are looking forward to organize such sessions on offline mode too. Thank you very much.
Thank you so much, madam. I would also like to request Dr. Dinesh Mishra sir to please present your views.
Thank you, Dr. Rajiv. Am I audible, Dr. Rajiv? So it's always pleasure to have associated with ISSRF. And since we got an opportunity to coordinate with Dr. Neelesh, I asked Dr. Neelesh, and he was very curious to join hands with ISSRF and to organize such a wonderful webinar. We discussed and we invited the eminent speakers from industry as well as from academics. So I hope the students have learned a lot in a whole day. And very soon we are coming up with offline session since it's just a beginning. So I request you all to join this society with not just as all the societies are there in the market. The thing is when you are really willing to work for the profession, since it's a society that is exclusively working on reproductive medicine, and we are from the pharmacy, so definitely we can join together. We can work on reproductive medicine also with the help of advancement of technology, nanotechnology especially. And you might have listened Dr. Shashank Singh and Dr. Tathagat Datta, Dr. Neelesh. Unfortunately, Dr. Bevo, he was engaged in meeting. But anyway, once again, I congratulate Dr. Neelesh and whole team, Dr. Rajiv and Smriti College of Pharmacy. And I would like to thank Dr. Lohia, President of ISSRF, Dr. P.K. Mishra sir, Dr. Ansari, Dr. Barkha, and all the organizing committee members and dear participants. Thank you very much. Have a good day.
Thank you, sir, for your valuable insight. Active participation and collaboration have reached this platform and foster the meaningful discussion. We are confident that the knowledge shared today will inspire further research and innovation in the vital field. Thank you all for your time, effort, and contribution, and we look forward to continue this journey together. At this moment, we conclude this as a remarkable, memorable, and knowledgeable event. Now we officially draw curtain on our today's National Webinar 2024 and declare the end of today's webinar. Have a great evening and please take good care of yourself. Thank you so much.